So as a note, we are not completely yet ready with AI or ML. Of course, there are a lot of studies, everything is getting implemented, success stories are coming, and we are yet to reach that particular level. So it takes time, like Human Genome Project. I'm talking about drug discovery. I'm not talking about any other area. In many other areas, AI and ML is already being implemented and it's in action. In drug discovery, we are still driving to reach there. A slide contains many of the contents and pictures or videos taken from web articles and lectures, and the copyrights is respective to the authors. I've seen that many of the participants were asking for the data. So I thought this particular slide will be available in my slide share. The link is given on the slide. And some of the hands-on, including Jupyter Notebook, KNIME, Padel, Qsar, Autodoc, Modeler, Dynamics, Molecular Dynamics, it's all in there in my YouTube channel. And today, whatever I'm going to use for the KNIME, Orange, as well as for Jupyter Notebook, I'll be using some codes for Qsar modeling and also for analysis purpose. They are all available in my GitHub. So it's all open. So you can go and find them once you're done with the presentation. So I think you already have all the data with you with respect to this presentation. So we are trying to actually bridge the gap. That is what we wanted to. I don't want to say about academia or industry here, but it's more about what is tomorrow's need, what is actually the market requires. As Professor Shastri was rightly mentioning, we were mostly focusing on publications and others, but tomorrow we want our students and scholars to be more productive, and employability should be there. There is a good increase in the percentage of employability when compared with the Times of India survey when compared to 2019. So in 2020, they say the upskilling has much better happened with the students on the employability criteria. But still, things are missing. So we need to upskill ourselves apart from your curriculum. We cannot say that everything to be added to the curriculum. So they have to look at what is expectations of market or industry or commercialization, or even why should we patent a particular technology, whether there is a need in the market. So patent means money. It's not only to put it in the CV, but it's tomorrow we want to make money with patent. That's what we are looking at. But when you're looking at the pharmacy, or when you're looking at drug discovery, we still have a lot of unanswered questions. There is no medicine for this. There's no drug for this. Efficacy is bad, toxicity issues. But still, as a normal civilian, they always think, what I would really like is something to cure the high cost of medicine. What can we do as researchers to reduce the high cost of medicine? Of course, tomorrow I cannot come up with a drug. It's not a one-man job anyway. There are thousands of people, researchers, regulatory mechanisms, everyone is involved. But still, the research has its own main counterpart with the cost related to the medicine. That's where we wanted to understand the failure. So this particular pie chart is being made to understand from the papers, both the papers which I have listed here, 1988 and 1997, where we are talking about how the oral drugs was failing because of what reason between 1960s and 1980s. It was mainly because of PK/PD or ADME studies or properties and efficacy. But when you're looking at strategy, toxicity, and adverse effect, it was bit minimal. Strategy, because we used human synergetics and expertise there rather than any much of tool, because that time we don't have too many tools or AI or ML, whatever it is, what we call, rather than more of statistics. And later come in between like 2000 to 2010. I don't have any data with 2010 to 20, so I didn't put it. So later we could see the trend has completely changed. PK/ADME efficacy is one of the least reason the cause of failure. The most reason was toxicity and strategy. So when you're designing new molecules, maybe using tools, or let it be any other sophisticated like algorithms, but still they were having issues on the strategy. So now we need to understand that there should be a good synergetic between human expertise and computational modeling. Completely relying on computational models No, that's not that great. So we need to put our expertise also. So science of relevance is something very important. So we have to also understand which is right, which is wrong, what is the significance of the parameterization, how to tune the particular parameters of a model. All these things we need to know. Without knowing that, we cannot simply use it. Someone used something, it worked for their project, might not work for your project, because your problem statement and your biological systems, your compound configurations is completely different. As Dr. Sunil earlier discussed about the bioisostereoreplacement replacement. When a replacement happen, you focus on a specific parameter. When you look at multiple parameters, you end up with nothing. So you have to go one by one and to try to consider and come up with an optimal balance. So estimation of ADME at early stages, where this is only because we could generate a lot of models to predict the ADME PK studies in later stage, that is 2010. But ultimately, we require experimental data to generate all these models. So similarly, for toxicity also, we require tremendous amount of experimental data in the following years to see the reduce in the percentage or the contribution to the failure of oral medicines. So today I'm bifurcating into four different topics. The first I will say about machine learning, what, why, and how. And then second I'll talk about the machine learning in chemistry, how the chemical data is being used in data sciences, and drug discovery on how to avoid failures. And finally, what to do next with, are we still ready with AI or ML in drug discovery? So, starting with the first part about the machine learning, learning denotes changes in a system that enables a system to do the same task more efficiently the next time. That's what our parents, my parent used, or everyone's used to tell. Okay, you made a mistake now. It's a first and last. Don't repeat that. So we want to be more efficient next time so that we don't repeat that mistake. The same thing. Machine learning means you are adding in data to improvise, to enrich the quality of the data or quality of the model. So we want to enable a system that to do the same task more efficiently the next time. So why do we need to learn? Because we want to understand and improve, discover new things like data mining, scientific discovery, and fill in the skeleton and incomplete specification. That is something very many of them are working on. Even my counterparts in UK is also working on data imputation. So data imputation means there will be missing data in many of the compounds which is similar, like assay data and all. So there are deep learning algorithms which will be able to help you to fill those missing data. Already some of the algorithms are there, but the applicability domain and the standard uncertainty was huge, but now we are able to overcome with that. So also build software agent that can adapt to the users to other software agents and reproduce an important aspects of intelligent behavior. So where the three parameters in learning system is, one is task T, performance P, and experience E. So how exactly what knowledge is to be learned, how this knowledge is to be represented, and how this knowledge is to be learned. When you see this, it is something that is related to human learning, right? Exactly the same. But we are transforming it to binaries. As in Dr. Karthikeyan's presentation, he was mentioning about the binaries, how the machines learns about what the data that you are giving. So that's only conversion happening. So artificial intelligence is something that we are enabling machines to think like humans. It has its own things that we have to take it into like understanding. But in machine learning, it's training machines to get better to do a task without explicit programming. So keep in mind, in statistic, we use a data, we come up with a model. But in machine learning, we can go on adding the data. It will learn itself and improve the accuracy of the model. And the deep learning, which uses a multi-layered networks for machine learning. There are hidden layers also there, so it could be a black box too. So that's all we have to consider when you are going for all these different variants. Now, currently in our life, AI is impacting on our smartphones, then detectors like the speed detectors, right, on the roads, then your social media platforms, e-commerce, they just understand what you search and they pop up with a lot of ads, right? Banking sector, finance, autonomous vehicles, and smartphones. And then machine learning is also a branch of computer science, which deals with system programming in order to automatically learn and improve with experience. Now, don't worry, you need not to learn programming as such, because we are all more into medicinal chemist or drug discovery guys, so we don't want to do too much of algorithm building. There are plenty of algorithms and libraries being built. We have to make use of it. That's it. So for example, you Autodoc, we didn't build, right? Someone has built it, scripts and all. We are just making use of Autodoc to predict and see the binding affinity study. So that's the same way we can do here. So the architecture, you have to understand that knowledge base, performer, critic, and learner. So critic is like someone is giving feedback about your work and you're improving it. That is a learner. So the problem generator is to test the performance against it. So performance should be checked at each stages. Today I'll be displaying during my demo how the performance is being scored and other parameters. And the paradigms of machine learning is that rote learning, interactive learning, induction, analogy, clustering, discovery, and genetic algorithms. I'm not going deeper into it. This already previous speakers have spoken about the different variants of algorithms like unsupervised, supervised learning, and reinforcement learning. Based on the data category and data types, we try to implement these kinds of algorithms. So there are main types of techniques in ML: supervising, unsupervised, semi-supervised, reinforcement, transduction, and learning to learn. And 5 popular algorithms are decision tree, which I'll be using today, neural network also, then probabilistic network I'll be showing you in the multi-parameter optimization, the nearest neighbor where you can correlate with your chemical structure and activity data. So how far your activity is deviating when there's a functional group is changing within your structure. So nearest neighbor methods can be applied there. And support vector machine also have used today for Qsar studies, so which I'll be showing in my demo. The stages of machine learning is model building, model testing, and applying the model. All 3 of them will be shown in the demo today. Then what is not machine learning is artificial intelligence and rule-based inference. So rule-based inference example I'll be showing you in the presentation. I'm not going to talk anything or give any demo about artificial intelligence. Machine learning is what we are more concentrating to. So let us see how machine learning has influenced or how it has helped in chemistry, especially in chemical data. So this was like a survey which was being taken by CNDN during the year 2018, where it was clearly mentioned what area of chemistry people are working in to understand whether machine learning is really overhyped or not. So you can see that around 31.8% agreed that it is overhyped, and from a response of 151 people, in which almost 23% was organic chemist, 16% was analytical chemist, 10% was biochemist, physical chemist 17%, inorganic 12%, but computational chemist just 7.3%. So we have to still see, because when computational chemist is more, I could see how this could change. But anyway, still worth of understanding in 2008. Now it could have, as of today, it could have absolutely changed. And the lockdown gave an opportunity to many of them to upskill with all these techniques and approaches. So one of the workflow in chemistry is that the chemist analysis, like they might analyze, they might synthesize. So there will be a lot of annotated data like sensor screening, synthesis, and simulation. And then we pre-process, because that is one of the important challenge when you're before going for machine learning models. You have to prepare your data and data types properly. So not simply clicking a button, it automatically does. No, you have to define the data types properly. So pre-processing is very important. So for that, you need to understand what is the kind of data and how to be applied in statistics. For example, when you usually apply any data to statistics, you normally normalize the data. We check whether there is a normalization required or not. The same way ML has also its own requirement for pre-processing the data. And then we apply machine learning models, what kind of algorithms to be applied, depending upon what kind of data type. When I say data type, it could be categorical or integers or strings. So we have to see what kind of data type will be useful. And then we generate the model. And then we decide about classification or regression. But advantage here is each time we add new model, the model gets, new data, the model gets trained again to improve. But again, we have to check with the applicability domain, performance, everything to be checked. The other criteria is that object-oriented data set. For example, we already have design of experiments. We do a lot of experiments and optimization. So in between, we make a model, a machine learning model, to minimize the number of experiments and minimize the material property to improve the what to say yield or productiveness. So we basically learn from the existing reactions and we try to optimize them, maybe any assays or whatever properties, and then based on the history we come with the proposal. Like Dr. Mandar was mentioning, the reactors, they also learn about the failures. Yes, it is very, very important, as you see in the second, this one. So framework for working with an in-house databases, history reaction. We need to understand failures as well as success. So people, students, whoever working, and if you have negative results, please include them to your literature, because that helps others to understand how not to work or what did not work. So this also helps the machine learning algorithms to understand which of the reactions was successful, which one was failure. So this will be always giving you recommended reaction based on different models. And then we do have data from simulations too. For example, there are plenty of simulations happening on DFT, calculation of energy. So if you do DFT, it might take longer time, but ML might be a little bit more faster, but model generation takes a bit more time. So here you can see the difference in the values, that is the energy values predicted by machine learning models and DFT calculation, that is quantum chemical calculations. The same, when you're coming to public databases, they use neural network to predict a reaction condition. For example, for this particular reaction, what would be the best catalyst or solvent or reagent or even the temperature for an optimal achieving the better yield for the this particular reaction. So all these algorithms and models are being developed, and many of them are openly available. Nothing is too much of proprietary, because still people are learning, so they want contributions from other people to improve the models. And these are deep learning algorithms for solving different chemical challenges, understanding the molecule, designing new molecule, synthesizing the new molecule, that is understand retrosynthesis pathway, and even predicting their binding affinity or energy values. So they try to learn and then come up with the predicted data. Similarly, this is a schematic representation of main component of atomistic ML. So when you come up with the output, you need to understand what is it about. This, we are talking about a scatter plot, but this is more of a clustering, or this is about a chemical space. So this is again a tree, decision tree kind of. So each of this representation gives a lot more interpretation capabilities. And this was recently in Kaggle, and it is also published, and it also talks about different data where they made a competition, and they have used different models to see what is the accuracy level. So AUC and R square. So you can see some models did not give that great R square, but some you can see the AUCs are even 94. So it all depends upon the data and the method that we have to look at. Both the DNN and non-DNN models were being used. This is the reference. So from a famous writers like Nathan, and also so he will be the right person, because there are many AI companies, as already been mentioned by previous speakers. So the descriptors for chemistry, how do we define them into the CPU so that they can understand how the chemical structures that is being transformed? For this particular structure, is easy for us to understand what is aromatic ring, pyrrole, or whatever it is, the keto group or oxygen, hydrogen, all those things. So the issues with machine learning is arbitrary size and arbitrary audio. But in ideally, general features are their general, compact, unique, invariant, smooth, and fast. The same way, this single structure is being represented by this particular binaries, bits and binaries. But when I'm going to go more definition, so local or atomic descriptor, I'm again defining each of this regions of the compound in a different descriptor. So these can be called as a fingerprints also. So fingerprints, so the ML models or even algorithm, they do understand a lot of fingerprints and numerical data when compared to when you draw it as a structure. Now let's go into drug discovery in order to look at the theme of this particular conference is also how to use AI in drug discovery. Drug discovery approaches are completely dependent upon availability of data. Availability of data means right data. When I say here 3D protein structure, I am looking forward to a good resolution 3D X-ray crystallography structure. I'm not talking about a homology model. Homology model has its own drawbacks. You have to make a lot of validation, molecular dynamics to ensure that you have the better structure with you. And then when I'm talking about 3D ligands, I'm talking about X-ray crystal ligands or the best conformer to start with. Because your ligand as such, when you synthesize, it has a crystal form when it got solidified or crystallized, right? But when it gets consumed and it gets binded to a biological complex, the conformation will change. So the biological complex conformation for a ligand or a drug molecule is different than when it is in a stable form. So our job in finding docking studies is to find out the stable conformation, which is stable within the complex. But the molecular docking, most of the time, we make our receptor as rigid. Now, that's where the importance of molecular dynamics comes into the picture. So we wanted to do molecular dynamics in order to understand how far your ligand is stable within that pocket for a certain amount of 200 nanoseconds or 100 nanoseconds. Minimum 100 nanoseconds is required, not 20. So that is a requirement. So that's what we call it a structure-based drug design. And when you go for fragment, we have a 3D protein structure, but we tried with all ligands, nothing worked. Then we come up with de novo or scaffold routing. The challenge here is we need to check out the synthetic feasibility and accessibility. So I always tell, if you're not a chemist who is doing this exercise, have an organic chemist or a medicinal chemist sitting next to you so that he or she will be able to tell you the best fragment that is synthesizable. Because unless you confirm that this is synthesizable, no point of making it in computers and predicting virtually. So we have to go for chance of success rather than missing the opportunities. And then very famous ligand-based drug designing approach, when you don't have a 3D protein, but you have activity data or you have a reference active conformation. Then you do a pharmacophore studies, similarity studies, even based on pharmacophore or structure-based or fingerprint-based, even QSAR also. Personally, I prefer 2D QSAR when compared to 3D. 3D has its own many things that to be taken care. So simply generating 3D structures are not viable. There will be a lot of false positives when you go for 3D QSAR. So you have to take much care about which 3D you're going to use it for 3D QSAR calculations. And when none of the data is available, you go for virtual screening, that is high throughput screening. So that's where we have to understand the role of docking. In 1894 or even in 2000, we talked about lock and key hypothesis. To exercise a chemical action, a ligand interacting with a protein must fit into the binding activity like a key into a keyhole. That's what we were learned. But right now, in 2020, this is only half of the truth. We cannot say a key lock hypothesis. Multiple keys can go to a lock. So that's how the hypotheses have changed. Understanding levels also have changed. I'll try to justify this. Why we are doing molecular docking, right? We want to determine the optimal binding structure of a ligand to a receptor, quantify the strength of the ligand and the receptor interaction. That's what we are looking at. Mainly, we are looking at where the ligand will bind. How will it bind? How strong it could bind? The role of solvation and desolvation. What makes a ligand bind to the receptor better than others? Translation and rotation of ligands. Torsions. Did I anywhere talk about docking score? No. Docking score is only applicable when you're doing virtual screening with considering the enrichment factor. If you're not able to talk about enrichment factor, reporting virtual screening data is of no use. We cannot validate that. But when you're doing a normal, let's say 4 molecule, docking score does not give you any sense there to rank the compounds. It's more of energy terms that we have to look at. So main of the key points that you have to look at is what is a catalytic function? What is the regulatory mechanism of your enzyme, which you have considered, whether it is an active site or an allosteric site? What is the cleft detection? Did you really identify the functional site? Did you identify the key amino acids which is there in your binding pocket? Geometrical analysis, is there an open or a closed conformation? Is there any change in conformations happen because of the binding of this ligand? Flexibility, whether it is on aposite or on a substrate bound site. Conservation, we are looking at selectivity, sensitivity, and specificity. Surface characteristics, whether it is a solvent accessible area, what is the hydrophilicity or hydrophobicity, and finally we talk about the interaction energy. So this is a picture that was taken from enzyme methodology book. It's really interesting that we have to have a better understanding when you're talking about molecular docking results. This is what I was talking about desolvation terms. We are looking at torsions of the compound, complexes, and then also hydrogen bonds and its contribution in log P. So we are trying to understand which of the compounds having or the conformation generated from docking could be present in a crystal database. So here you can see the green and yellow and orange. So the color represents that if it is green, it observed in a Cambridge Structural Database, that is CST. So if this particular dihedrals or torsions are presented, that means this particular biological conformation is possible to crystallize. So that's what we are looking in terms of. So where is docking score coming into the picture? I said docking score is very important when you're going for virtual screening. But ultimately, as a medicinal chemist, you do not want to simply do virtual screening. You wanted to synthesize new molecules. You want to come up with the lead molecules or derivatives. That's where we have to look at. Another one is that this particular carbon atom is having a negative contribution to the binding affinity. So I should think of whether I can come up with some new derivative or replacement of this element to see whether it enriches my affinity. That's what we have to look at, the contribution when you do a lead optimization on a 3D based lead optimization using your protein structure data. And finally, we also look at chemical spaces. There are a large amount of data. Last week, there was ultra huge, large databases where being chemical spaces. There was a workshop from NIH. So there they were talking about huge databases, like billions of compounds in a databases and all. So there are many being spoken about, even people from Merck, AstraZeneca, Pfizer, even from Google. Many of them were talking about the expansion of this chemical space of data. So this is going to really help the medicinal chemist to understand what could be the best building blocks to come up with novel compounds based upon the pocket of your protein. That's what is the revolution. So they try to do a mapping, not simply based on a structure similarity, but more based on a pharmacophore similarity. So for example, this particular region is being made similar on the pharmacophore concept, and they found this to be similar. And you can see the similarity is 0.61. And this one to this one to this one to this one. So pharmacophoric features are being found similar. So this was our query molecule, and they were able to find a new virtual hit based upon the building blocks of chemical space based on the pharmacophore criteria. Now the global similarity will be 0.8, but I don't think when you do a structured similarity between this compound and this compound, you will get 0.8. It will be lesser than that. So that's what the mapping works actually. And also a lot of applications of machine learning on retrosynthetic pathway, where IC-Synth also being used. You just give a new compound, it will be able to fetch the database and then look at the literature and predict the similarity and the yield, what could be the strapping agent, and all the data could be predicted. So finally, the objectives of drug discovery is we have to come up with an optimal balance of property. When you do a molecular docking or a bioassay studies, you're just finding a hit molecule. That's it, where we understand the potency. We never could call it as a drug-like molecule even. To call it as a lead molecule, you should find that your drug is safe, at least some extent safe, of course, on our level of research. And then good amount of absorption rate in human intestinal absorption. Metabolic stability, my drug doesn't break down, or when it passes through cytochrome P450, it is stable. Solubility and flexibility of my molecule. All these things to be considered in order to call them as an optimal drug candidate rather than a drug molecule. So the three mantras are like fail fast, fail cheap. Only when confident you move forward with that compound, and avoid missed opportunity. Do not use unnecessary filters, which is not really required for that kind of objective, so that you might miss opportunity. This is only because there are a lot of data. The data overload is also a big challenge. So if you let's forget about what is written there. Rather, I'll give you a very simple example. Let's imagine you have a 10 different compounds. In order to identify two best drug candidates, I have to carry out 10 different experiments. If I am doing one single experiment for the 10 compounds, it is easy for me to pick up the best two based on thresholds or some criteria. But here I have 10 different experiments, independent experiments to be considered in order to choose the best two from the 10. How to do? Most of the time I hear from audience and participants and researchers that take an average. But in my question, I emphasized it is independent experiments. All are important. So we have to give priority. So ultimately, what we have to understand is, I'll come to the point. We have a lot of data from in silico, in vitro, in vivo. And then how do we come up with a conclusion? We have to prioritize. We have to give importance, which properties or which experiment is more important than the other for this particular objective or activity or property that you are looking at. Second thing is, you have to consider uncertainty is also. Uncertainty means error. Or when you say a single experiment, you carried out experiment just once, there is uncertainty. You don't know standard deviation. There's no mean. At least 3 times you have to take it, right? So when experiment itself has uncertainty, when you come to prediction, of course there should be uncertainty. So we need to understand the standard deviation of the prediction also. If there is a more deviation, we don't give much importance because it is unreliable. That doesn't mean that the software or the model is bad, because the chemical class of the compound that you use for prediction is not within the applicability domain of training set. Sorry, I know it is might be a little bit vague, but you have to a bit understand about applicability domain so that you will understand more about Qsar and others. And finally, when you do selection, the selection process is not using a tool. You have to do manually, sit down, analyze the data. For me, initially, when you do a selection of compounds from your big, large set, you have to give more importance to diversity and then quality. So don't just pick up a single molecule and just build up the derivatives. Rather, have different scaffolds and then have multiple derivatives, and that's where you have the diversity there. Then you have a quality. Later, you can give more importance to quality and then diversity. So that's how the process should go on. The initial conventional filtering, what we do is we have set of molecules, we do for potency test, and we remove some of the compounds which might be not that active. And later we try to carry out absorption rate studies for industrial absorption. Again, we remove some of the compounds which does not work. And then again, we expect that some of the compounds will be really nice and good. Then we did a metabolic stability studies, and again, we removed some of them. And finally, we expect that we are getting a very good compound. Unfortunately, in most of the case studies, we found that, or reported studies, we found that this is not happening. Your best compound will go and hide somewhere because it might not be having a good absorption rate, even though it had a good potency. Or the same case you might have experienced, even though your compound is very active, you found later it is toxic, the same way. So you might end up with a non-active compound also. So when you do step-by-step filtering, there will be chances of missing opportunities. That is what I call missed opportunities. So we have to think of how to apply different methods. So this is where we are using rule inferences, rule inference based method. This is not machine learning, but this is rule based method. So where we try to consider different properties, and also we try to give the threshold, and we give the level of importance, how far this property is important when compared to the others. Something similar to Lipinski rule of 5, where they have said one rule can be void, but others should be sufficed. So the same, similarly, some rules are being come here, and it is being measured on probabilistic scoring. This is already being tried. This is a published work where they could find only 60% of Pfizer candidates when compared to applying multi-parameter optimization. They found around 74%. So the difference is 14% was a missed opportunity when they did it one by one. And again, ADME, we are looking at the distribution, metabolism, absorption, excretion. I'm not going too much into the theoretical factors, but all the ADME models are based upon QSAR. We need to have the right starting structure. Mostly it will be 2D, not 3D. And then we extract the descriptors, and we get the data of all these ADME properties from experimental assay, and then we call it them as a response variable. And then we apply it on different statistical or machine learning models. I remember 2014 or 13, Dr. Sunil was one of the guest editor for Bentham's medicinal chemistry. And then he has inviting for what to say, articles. Myself, my professor back in Professor Matti Carlson, we wrote a review on machine learning in drug discovery. I think back in 2013 or 14, I think. It was published in Bentham Journal of Medicine Chemistry. So that's what I remember, because this was a particular slide I prepared for my PhD. So still I'm continuing using it. So that's what I remembered now. So finally, after generating all these models, it's not just simply about R square. It's not just simply about less error value. It's also about physical meaning, significance, relevance. So whether your property or the descriptor that you have chosen has a good relevance to the activity or the property that you have chosen for modeling. That is very, very important, even though statistical parameters are also equally important. So finally, you have to do validation, and then finally you go for prediction. So ultimately, you're generating these QSAR models so that others can use your model and predict the activity for their compounds. For example, pass server, that is what they do. So whenever we publish QSAR paper, ensure that you come up with all this raw data dumped somewhere publicly so that people can use your model to predict their activity. Otherwise, it's of no use actually. Now, today we will be looking at the demo session on generating random forest, then SVM models also. And we will be also, I won't be dividing today's set to validate, but we will be dividing only into splitting into training and test set only. So we also have a validate or test with independent set in order to understand the applicability domain of your model. This is what is called domain of applicability. We want to understand the diversity of a training set which defines a domain of applicability. For example, let's imagine these are the orange dots are the structures with similar clustered to understand. Now, I bring a new compound in order to predict the activity using this training set, which is the model I generated. If my compound is fitting within the applicability domain, the standard deviation will be less. I have more confidence in the reliability of the model prediction. If my structure is slightly away from the applicability domain, okay situation. But if it is too far away from my applicability domain of my training set, it is unreliable because the standard deviation is more. So I cannot take this prediction as a validated one. So unless a QSAR model or a ADME prediction comes up with standard deviation, the reliability of the value is questionable. How do you like justify yourself this is a accurate data? I don't say accurate because you cannot predict any accurate values in models anyway, but at least reliable data with more confidence. That's what we have to look into it. Now, some of the challenges I already told you in deep learning, where imputation. So traditional QSAR modeling offers a little advantage, and the compound bioactivity or property data is also very sparse. So we want to add this missing data, or we want to work on these experimental data to make better prediction for compound bioactivities and all. So let's imagine you have a descriptor and you take the prediction and come up with the prediction. But let's say you have assays, but there are a lot of missing data here in the assay. So we can use a deep learning algorithms to impute the assay data to it. I'm not going too much of the details here. A lot of publications have come up and case studies. So this will be a bit more lengthier talk, almost 2 hours of talk. So I don't want to go. So, but it's about, we are understanding about the descriptors and the assay. When we combine together, we are able to impute the missing data available using deep learning methods. And also imputation is to check with the probability with the predicted values. And these are the different applications being as a case study, which already being worked out with several pharma industry, also with the open source malaria project, where they are able to get with generative models. So ultimately, what we are trying to understand from this drug discovery section of my presentation, we are trying to bridge the gap between a chemist and a modeler. Whatever you model tomorrow, if it wants to say that this is working, a chemist have to synthesize this and to get it tested. So there should be a good synergetic and a collaborative ongoing with the chemist and the modeler at any region. Let it be pharmacologist or pharmaceutical chemist, whoever it is. And if you look at the success stories, these are the list of success stories. And I think one of the review was written by Dr. Sunil. I have to check. I'm not sure. But These are the main reviews from where this list is being prepared. So there are good success stories of CAD and ML, which has gone to phase 1, phase 2, phase 3 of clinical trials. So we have to wait and see of more news coming up. Now how this machine learning can be applied, how we can do it ourselves. So that's where we have to simply depend upon a single particular package called as Anaconda. Anaconda includes all Jupyter notebooks, SciPy, NumPy, then NumPy, TensorFlow, Matplotlib, Pandas, Scikit-learn, and many more Python libraries. So you have to just install Anaconda. That does the job. So today I'm going for the demo now. I think I have time. Yes, I do. We will be doing a SAR model using machine learning methods. One is SVM, random forest we'll be trying. So for that, we are using RDKit, which already Dr. Sunil has already mentioned it. So RDKit is a collection of cheminformatics and machine learning software. Then we will be using Scikit and Pandas. Scikit is again a free software, especially for machine learning libraries for the Python. It features various classification, regression, and clustering algorithms, including support vector machine. So it is already a library is there. Only thing you need to know how rightly it to be used, because there are specific syntaxes where you have to define it. That's all, because you need not to write lengthy codes and also not at all required. I am not a programmer, so but still I'm able to make use of Scikit and Pandas and all. And Pandas is a software library for data manipulation and analysis. It offers data structures, operation for manipulating numerical tables and time series. For example, I told you earlier, before doing machine learning analysis, you have to treat or pre-process your data. So Pandas libraries can be used for that. Another thing, you hate completely coding or anything like that. I don't want too much of flexibility. I'm just a graphical user interface guy. So then you have two options. One is Orange and KNIME. KNIME, I have already published many of the workflows in my GitHub, and there are a few YouTube videos which I have already given for cheminformatics. So today I'll be showing you just quickly because of time limitation. So let me go to the first demo of KNIME. Yes, I hope you can see my screen. So let me tell you about the different parts of KNIME. KNIME can be downloaded from knime.com, and it is free, or knime.org, I think. And it is a free open source software. The desktop environment, the server environment is commercialized. You will have a lot of nodes available here. Just a minute. Yeah, so you have a lot of nodes available here. So these nodes can be installed. How this installation is happening, you have to go to help menu, install new software, and you can able to install even many of the free nodes. Then you can install Biosolve IT nodes for docking, then ChemAxon nodes for Chem Sketch, Chem Viewer, Marvin Sketch and Marvin Viewer, not Chem Sketch. So these are already free, and many of the RDKit and Indigo and Enilos, Earlwood, then Padel, Veka, 3D Chem, and CDK, many more nodes are available. Now, what is the use of KNIME is automating the process, but be careful with the automation. You have to test it, properly check everything, and then only automate. So what I'm saying automating is, let's imagine you want the software itself or the KNIME itself to download the Chembl database, do pre-processing of selection of compounds, neutralize, desaltination, that means remove salts, and then you want to convert 2D to 3D. Then you want to generate conformations. You want to do docking. After docking, you want to do some clustering. After clustering, you want to do some pre-analysis and post-analysis with conditional constraints. And then you want to come up with pharmacophore. Whatever steps I told you, you might be using different tools, and you have to wait for each software to finish the calculation and all. But in KNIME, each of the nodes, you can define it and then connect them automatically. When the first collection or downloading of the compounds finishes, automatically the next one will start. And you can just, once you optimized and checked or tested all your workflow, you can just click a button, it runs it automatically. And once at the end of the day, when you finish and come back, you will see all the results. So that is the advantage of KNIME. I've been using KNIME since my PhD days, since 2011. but that time there is not too many nodes available, but now you can see lot many nodes available. So let me explain what is this workflow is about. So I am getting a database using SDF reader, and then I am drawing a structure with ChemSketch. Okay, what I'm doing here is a similarity study, substructure search. So I already have a SDF. When I double click on it, I have option to load the SDF file. If you want to see how many molecules are already there, I can say scan files. You can see 191 molecules are there in my SDF, and these are the three columns: compound ID, PKI values or predicted KI values, and series. Okay, now that's okay. I didn't change. That's why it won't. And then ChemSketch. I'm double clicking on the ChemSketch. It will pop up with the ChemSketch drawer, drawing tool, and then you can draw the substructure there, so that you are saying that I want to search that just this aromatic ring, or if I want to give some other thing, I can do that. And I just click OK. Since I didn't make any changes, it is saying. So it will generate the fingerprints of all your database. I just loaded only 191. You can load millions of compounds based upon your memory of your computer. In KNIME, it is not about more CPU, but it's more about RAM and cache. So you need to have a lot of RAM to work on KNIME. And then you generate the fingerprints of your compound, and then we try to do a fingerprint similarity search. So one substructure against 191 molecules, and then we generate a distance matrix calculation in order to generate the final similarity viewer. So let me run it. So in order to run, I hope you can see my cursor. You can click on the top. There is execute all the nodes. Otherwise, if I just select this, I can run just that particular node by clicking the single play button. Otherwise, if you want to see what are the SDF that is already read, so here there are traffic lights. If red means data is not sufficient or not given. Yellow means data is given but not executed. When it turns to green, that means all accomplished, result is ready. These are the output nodes and these are the input nodes. You click the right button and say red molecules. Click on red molecules. And then it should show a table, which is something similar to Marvin view, and you can see the 2D structures and what is the content that is read by the SDF. Okay, now since we have read, I'm just clicking on this so that all the will be executed. So see, it automatically got executed and the final one is running. I'll show once this is finishing. I'll show the output. It looks really nice because it will do a comparison between a substructure search to find out the similarity with this particular database, or 191 compounds. I cannot say it as a database. Yeah. So I click again the right button and I say view the heat map. So the heat map, it is the advantage here is it looks colors, but what I'm going to get information. Just mouse over on top of it. You will see which compound within the data set against the other compound which is having more similarity. If I go to the middle, of course, they're exactly the same. The similarity is 0, means they're duplicate. If I am going above, you can see the similarity is changing like 0.5357 and also this also represents, if you click, you can identify the row numbers which will tell you which structure to which structure is similar. So when you do QSR, it's always better to understand about the homogeneity of the homogeneous compounds or data set. You can always check this. Now let us look at the other one, similarity to target, because my aim was to do a substructure search against the compound that I drew in the ChemSketch. So it found, let us see. So here I have row 0. I have to see where is my, I think row 0 is my structure, or maybe I have not added it. Sorry. Yeah, I have to add that. So here what I'm doing is I'm selecting a structure within the data set and I'm asking how far it is, how this row second structure is similar to the other compounds. So this is sometimes I really wanted when I wanted to do any QSR, any studies like that. Okay, so similarly, if I'm going to the second workflow, I'm not going too deep because of time shortage. Here what I'm doing is I am again trying to generate 3D structures. For example, I read the 2D structures of SDF files, and then I removed the small fragments or salts from my compounds. I added hydrogen, and then I did a optimization using force field like universal force field or MMFF94. And then if I want, I can generate again 2D if required. Otherwise, I can use this for my docking studies. Or the other way is I can use my SDF reader. I can simply use OpenBabel. So OpenBabel I can give to generate 3D also. So I have to give minus minus gen 3D. It will. So if I say here optional parameter gen 3D, it will generate 3D structures, and all the input files will be supported. Whatever you do in OpenBabel, the same thing can be used. Only thing in Mac, it don't come with the Babel binaries. You have to separately install and mention the location. Otherwise, in Windows and Linux, it works by default. Now, another one where I have done a Qsar modeling, which I have taken from the workflows from KNIME website. So, but I have added something extra. So here they have used a random forest regression method. We use SDF reader. We generated fingerprints using RDKit, and then we partitioned it like a test set or and sampling, and then we generated the model. Using the model, we predicted it, and then we generated the scatter plots and all. So this is again can be done within KNIME. So many applications are there within the KNIME. Now in this particular workflow is to generate the descriptors. So I can generate descriptors using CDK, XLogP, RDKit, NLOS molecular properties, then also Padel descriptor. So I know many of you will be using on Padel descriptor. That also can be used here. Advantages: I am just loading my single SDF reader. All softwares will run together. I can merge them. Finally, it will write it as a CSV file. So this is one of the best advantage with KNIME. Next, I'm going to Orange. So Orange is equally something similar to KNIME, but not many things. But in Orange, you can do only something similar to machine learning and statistics. You cannot do a lot of chemistry there. So what here I have done is I've taken a log BB data, blood-brain barrier data, and then around 56 structures are available. I have loaded this in my file. So this is what I told you. You need to know what kind of data types they are. So here you can see the name I have given as a number, and that should be a feature. So for example, descriptors are all my feature, activity will be my target. And what is a category type? Is a categorical, or I mean continuous data or numerical data. So at the end, I have an endpoint where I have already mentioned it as target. So that is where my target point. If you want to visualize them again, you just double click this, you will be able to visualize all your data here. Okay, this file is also available in my GitHub under Qsar. This file is there. Just download Orange and load that file, and you are free to change this CSV file. You can generate Padel descriptors, include your activity data there, and then you just load it here. It does all the modeling automatically because it's already there. Now, I wanted to select specific descriptors. Basically, I don't want to select all the descriptors. Too many descriptors, false positives, all overfitting issues, all those things will come. So I don't want to select many descriptors. I deselected some for while testing and then this I'll come to later. So I wanted to generate model using neural network, support vector machines, random forest, and tree, right? And then finally, I wanted to generate the models. So that is under test and score. When I double click on it, it will show you the results. I hope you can see the screen. So here it is telling me R square is high for tree, 0.95 for SVM, 0.87 for random forest, 0.99 for neural network. In fact, I won't take neural network anyway, because the sample data set is very less, only 59. So other one is I can do a cross validation. When I click on it, it will do a 10-fold cross validation. Calculations will be running. Here the particular window is covering up. Otherwise, it will show the percentage how far it is. Like the calculations are running. I cannot see. Let me check whether. Okay, I'm not able to see it. Just a minute. Okay. Yeah, it is calculating. See, you can see timeline is going. Let it finish. We will wait until then. And after that, if you want to make predictions, for example, how these models have predicted my experimental value using these model, I can put predictions, and then it will generate the prediction. So how do I get all these? Under data, you can find file, CSV file, and select rows and all. And under model, you can select SVM, neural network, and all those things. Now let's imagine I made a model. I want to share it with my friend or a collaborator who wanted to use this model to predict for their compounds. So you can save the model here. So for example, you see here, so I'm saving the random forest model, right? It will be saved as a file, and your friend, your collaborator, can load that model as a file again here, and then he or she has to load the descriptor, just the descriptor without any activity data, just the descriptor files of his or her compound. And using this model and using the descriptor, it will be able to predict the activity of the new set of molecules. So in my presentation, I told you building the model, testing the model, deploying the model, or applying the model. That all three is covered here in this particular workflow. Even in KNIME also I showed you building, deploying, or both, always I have showed you. But here I am giving you a little bit of saving model option. In KNIME also you have save model option is also available. This I thought it will be much more straightforward, so that's why I thought to more focus here. So let it finish, then I will show you the output data and prediction. So this is doing what to say, cross validation using tenfold. If I would have given 5, it would have been much quicker. So ensure that you have a little bit of RAM and CPU available, because that's why I said model generation will take some time. This is only 56 molecules. Imagine when you have thousands of molecules, so it will take some time. But here you are not doing any coding. So let me double click and see. Yeah, I can see it's not that great, but it's okay. At least I want to show the results, right? Now let us see what is the scatter plot. So I can see the scatter plot here. So in the scatter plot, I can define which model I have to use. The predicted model, I have random forest, SVM, and neural. Let me check neural network. And this is my endpoint, the actual experimental endpoint. If I want to change the color, everything I can do. If I want to save this picture, at the bottom, there's a save button. I can save the image. You can change the color, increase the symbol. All things can be done within here. Okay. Okay, so that's it with Orange. Now I'm quickly going to few more things. So in order to open Jupyter Notebook, you have to go to command line. After installation of Anaconda, you have to load Jupyter-Notebook. That's how the Jupyter Notebook will load into your browser. Then before that, the same slide, whatever I'm showing you, is already there in the slide share. You can view them there. And today, whatever I showed you this is the GitHub, GitHub/GitHub. So if you go to Qsar, whatever I showed you in the orange, the file is already here under orange, and you can download this file and just load it to your orange. So it will be there. Right. Otherwise, if you wanted to work on more Jupyter notebooks, I have Jupyter notebooks included for GROMACS, which does molecular dynamics. I am working on Amber, OpenMM, as well as for umbrella sampling. It will be uploaded soon. And then whoever wants to do protein modeling, already the advanced and basic modeling of modeler tool is already there, and it has given instructions how to do. So you just require your initial sequence of the protein. Rest of the things are literally automated. Please read the instructions and go. Now let's go to the application. So I will go in the present mode quickly, so that it will be visible much better. This is a Jupyter notebook, just a fun experiment. Voice recognition or typing script, because I don't want to go for too much science, then it will be a bit boring for you. So what I did is I am importing Python audio, Google Translator, speech recognition, and PyTTS X3 libraries into Python. And I'm importing those libraries called speech recognition and Google Translator. And then I recognizes the speech recognition. And then I'm asking the code, the Python code, to identify what all mics I have. It identified. And then I selected my mic, which I have connected right now. And now this is the beauty. So this I earlier said, but now I'm going to execute it again. Okay, let us try this experiment. Hi everyone. I'm very happy to be presenting my slides in this particular session. Wishing you all best luck. Yeah, the time is up. So I'm very happy to presenting my slides in this particular section. So I just said over the voice, the Python algorithms identified it. The Google Translator helped me to recognize the speech and translate it to the particular text. So see, we thought this is a big science or big coding required. No, now things are much simple. Just six letters of code, 6 lines of code. I'm able to um, uh, transfer my audio into text. See, this is the advantage. So you will start liking it. So start doing with fun experiments. Don't do science at the first, because initially you need to get attached and get more interesting to your Python programming. Later we can also do translations also. So let me go to science a bit more. This is the fun experiment is done. So this is again, I'm going to Jupyter Notebook again, where I'm using QSR blood-brain barrier, which understands the permeability of compounds. So this was adapted from an open source, openly available, published notebook from Maria and Pavel. So I want to import the library. What are the important libraries, which I already showed you in the presentation? We need RDKit, NumPy, scikit-learn, and joblib is also was part of scikit-learn earlier. Now you have to import it separately as joblib. And then we have to also import RDKit and others, right? Now I am reading the molecules and activity data from an SDF file. It can be any SDF file. 2D, especially not 3D. So I am loading it from, which is stored in my data logbb.sdf. I wanted to read one by one, and I wanted to also define it as logbb class. And then I wanted to see how many of them are there. There are around 321 containers for activity. That means the data features are there. Now, if I want to visualize the data, for example, this is a 24th molecule. Let's see if I want to see 45th molecule. This is the 45th molecule. If I want to see the 4th molecule, so don't think that if you just give moles for there, you will get that. You have to do the previous steps, which I showed you. Otherwise, it will not work, right? So this is how you can visualize the molecules. And next, this is something very interesting. I remember a previous speaker was trying to show, but I think sometimes it doesn't work. Sorry, even sometimes the lot of dependencies are there. So NGL view is a very interesting visualizer within the Python libraries, even in Jupyter Notebook, where you can visualize your trajectories of molecular dynamics, even proteins also. So this is a particular GROMACS file, which after generating a confirmation, which we can view. So you can zoom in, zoom out, all those things can be done. Trajectories, movies can be generated, anything. So just want to show you that, otherwise we'll go back to our work. So now we want to calculate the descriptor, especially fingerprints, using RDKit. So we are using RDKit and we are especially using Morgan fingerprints with a radius of 2, and we generated the fingerprints. After generating the fingerprints, we will go for converting the fingerprints to 2D NumPy array. So again, I don't want to confuse you with too much of things, but we are converting to the array so that the models, the machine learning models, can understand them properly, right? And we understand about 321 to 2048 features. Now we want to split the whole set to training and test set. That's the exercise for QSR anyway. So we try to split them. So here I have given test size is equal to 0.2. That means 80% of the data set will go to training and 20% will go to the test set. And then I'm trying to display how many training set I have. It is around 256. And then I'm going to scale them, because this step is very crucial, because in case of binary fingerprints, it may be less useful, but for SVM, it is very crucial. So we try to scale them to the standard scaling. And then okay. Here also we can dump the model. So you can see the dump scaled model can be saved as a PKL file, and later we can call this PKL file for models. Now here we are doing cross validation, exactly what we did in orange. There I did a 10-fold, but here we are doing only 5-fold. So that is what n splits equal to 5, random state as 42. And then I'm printing out what are the how many folds. So here, if I scroll, let me see if I can do that. So, okay, I'm not able to display that. Then I will be generating random forest model by giving estimators. So this is very important because we don't know, we want to fine tune it. So optimal tuning of parameters are very important to build the models. So these we want to understand which n estimators will be best optimal for to generate a best accurate model. So those things we have to define. We have to understand about RF modeling, SCM, SVM, and all. Then we did like a model building has been done with iterations, and finally we did a five-fold fitting to understand the model. Now let's save the model, which I told you we can save it as pkl, and now let's analyze the model. We want to predict the model. We want to use a model to predict the test compounds, which we have already split it, and then calculate its performance. So now we are loading the model, which I already told you. The scale.pkl is a model which we can load it and predict for the test set of molecules which are already bifurcated, which is 20%. And then we are predicting the values, log BB values. After the prediction, we want to check how far the test prediction came. Now you can see the accuracy score is 75%, 75.38%. So 25% compromise. So we have to still optimally look at tuning the parameters. Then correlation coefficient is around 49.95, and kappa score is around 49.85. These are all statistical parameters. Next is we are looking at the estimation of applicability domain. As I told you, QSR, if you're doing 100%, you have to report applicability domain. So we want to predict the training set's applicability domain, and then we are building up the SVM model again to check the process. So earlier we built with random forest, now we are building with SVM model. So the accuracy, the performance, things will completely change. So that's where we are trying to predict and understand how the threshold. Here you can see the accuracy has improved around 77.7%, and the correlation is around 54.87, and kappa is about 54.2. So there are more models. I'm not going there because there is limitation of time. So let me finish my balance slides, a few more, 3 or 4 more slides, and then I will wind up the session. So we are done with the demo. So we covered Orange, the similar things on KNIME, and the similar things on Jupyter Notebook. Now it is your choice which IDE or which interface you wanted to start with. So don't think that only a programmer can do all these things. No, never. Please spend some time on YouTube. There are plenty of videos for Jupyter Notebook, KNIME, and Orange. You have to spend some time. So even during your free hours, if you're really enthusiastic to getting into machine learning. I don't call AI anyway, we are not yet there. Now, what to do next? Are we ready for AI and ML? When you're looking, because I'm talking to NIPER, and I hope many of the faculties are related to pharmacy, we do have something called as Pharma 4.0. So where drug discovery is more of cloud-based simulation, model outcome testing. That's what we did it. We want to test the performance. That is very trivial nowadays. Better accuracy, better goal. So social collaboration with stakeholders, mobility-driven real-time electronic research data exchange. That is called lab notebooks, electronic notebook. In academia, it's not much used, but in industry, it's very widely used. And clinical and preclinical trials on electronic data capture systems, pharmacovigilance, clinical trial data, and many more. And supply chain, you come up with RFID, manufacturing, marketing, sales, blockchain, many more comes into the Pharma 4.0. But this is all under the Industry 4.0, where we are today talking about advanced robotics, 3D printing, Internet of Things. For example, Dr. Karthikeyan is more working into Internet of Things in biological problems, like yesterday he was discussing about cancer disease and his applications. I have tested his system anyway. I just send only the PDB ID and the molecule name. I just emailed it. After a few minutes, I got the result back with the scores and all. And he just called me to confirm whether you received it. So it was really great, actually. So that's how it is Internet of Things, actually. Artificial, nobody is sitting there and doing, it's all automated in the machine. So artificial intelligence, big data. So intelligent, flexible, distributed production. That's all the era that we are looking at. But the digital transformation has its own drawbacks, its own limitations. So we need to know where we cannot replace humans. Many places still cannot be replaced. So many of the times when I present this slide, people used to ask me, is it a danger to humanity, or is our job at stake? No, never. It is maybe for a short time. Our jobs might be in stake for a short time, but later they require them us to work, because data generation cannot be done using computers, only by humans. So this is one of the consortium. They told us, I don't know, it is official, but it is already, they have a website and everything, so it is taken from internet anyway, not my claim. So artificial narrow intelligence, which is what we are today, whereas job is enhanced. I could know that there are more than 93,000 job openings in India, especially on data science. But tomorrow that is going to go down, because once all the models are up and very efficient, then we might see a little bit down on the some of the job, not all jobs, but still again it will go up, because still they require it. So ups and downs will be there when technology implementations are there. We were all being worried when Y2K came, but did something happen? We just worried only on 1999, 2000, that's it. From 2001 we are back to job, a lot of job openings. So that's how it is. So all these technology implementations will have a little bit dip in the job openings or at stake, but later it has to boost up, because a lot of manpower is required. But I don't know whether this proposal or perspective that they are looking at 2040 and maybe beyond that, whether this will be, don't have any claim, and I don't take any stake here. So it's all we have to wait and see. So that's all from me for today. To conclude, when you're doing any kind of virtual high throughput screening or large data set screening, initially give importance to more of diversity. I'm talking to especially to medicinal chemist, and 25% to score or quality. And as the size of the lead reduces, the funnel goes down, you consider more importance to the quality or the score and less to the diverse. There you will have more derivatives rather than diverse set of scaffolds. Please consider enrichment factors when you're doing virtual screening, very much mandatory. And good synergetics between human expertise and computational tools are very important. It's not about which computational tool you used. No, it's about whether you have valid results using any tool. When I published these many papers, nobody asked me why I didn't use any commercial tool. I have used Autodoc for most of my publications. For QSR, I have used Padel, then QSRNs, which is absolutely free of cost. So nobody asked me whether you require any, but it has its own. We have to justify ourselves whether my model's performance is good or bad. So once you come up with these models, you will be able to prove yourself how far your model is reproducible. Avoid missed opportunities. Try to put the right method in the right place. Do not misuse it. Understand significance of parameters and properties. For example, there I told you, when my test set is 20% and my n estimators in our random forest is 100, 250, and 500, this accuracy is only 74. If I fine tune those parameters, my accuracy might be improved. I have to check that. So we need to understand what is the significance of changing that n estimators in random forest to get the influence on accuracy. Evaluate and decide the tool or approach. Don't think that pandas or skykit or rdkit can be used everywhere. No, it has its own limitations. So we have to test it, evaluate it, and make the decision which one is the right choice. And check reliability of the data use. That is very important in machine learning as well as in any science in drug discovery. Until unless your initial data is very well validated and from a reliable source, it cannot be understood in a proper way. One more thing is, when you're doing QSR, I have heard from many students that scholars, they try to merge data from different papers, maybe from same group or from different group, but ensure that do not do that. Do it only if you are sure that the assay that was carried out in order to calculate that biological property or activity is exactly the same with all the papers you have collected. If there is a difference in assay, don't combine them. There will be outliers, there will be deviation more in your, so there will be more false positives. Earlier days QSR was mostly used for local data set, not for very diverse data set, right?